Hello and welcome back, friends. Welcome to another video from Shomu's Biology. And in this video we want to talk about the coronavirus vaccine update. Regarding the coronavirus vaccine, how many days to go, how many days we need to wait to get a vaccine, and about the different aspects of the vaccine development, how we are going to develop that. We have already made a video in Hindi version and I've got tons of requests to make it in an English version, so I'm making it in English version this time. So stay tuned and watch the video to understand what vaccine is and how exactly we are going to prepare a coronavirus vaccine and how close we are to get a coronavirus vaccine. Okay, so let's start talking about the coronavirus vaccine. So like every single type of vaccine, if you want to know what vaccine is and how vaccine work and how vaccination work, I will recommend you to watch one of my videos regarding what is vaccine and what is vaccination. And there are plenty of videos of vaccination in this channel that you can watch for general audience as well. But for simplicity, let me break down what vaccine is. Vaccine is a substance that helps to develop our immune system, or that helps to boost up our immune system to fight against any infectious agent, whether that is a bacteria or that is a virus. In this case, corona is a virus. In this case, the SARS-CoV-2 is a virus, and this virus has a particular structure of a membrane, and surrounding the membrane it has an envelope, and then there are spikes coming out of the envelope. Okay, this is how the virus looks like, and there are multiple proteins embedded. So spike is one kind of protein. Apart from that, there are other proteins embedded in the membrane as well, which are known as the membrane proteins. And apart from that, it also contains its genetic material, which in this case is positive strand RNA. It's known as plus single stranded RNA as a genetic material. You don't need to know about that. I'm coming there soon. So this is known as S spike, okay, spike protein or S protein, and these are some other membrane proteins or M proteins. This is the overall structure of the virus. Now the thing is, a vaccine, as I told you, it's a substance, it's a particle which helps to boost our immune system, boost our natural immune system to fight against infection. So what we do and how you prepare a vaccine. To prepare a vaccine, the most important job that we need to do is to take a portion of the infectious agent, which is a virus or which is a bacteria. In this case, a virus. So take a portion of that virus, and that portion needs to be injected in our body in order for our immune response to respond against that particular portion of the virus and to generate antibody. Okay, antibody are specific proteins that will target a specific protein of the virus. Now these antibodies that is present in our body, normally there are basic antibodies present which are non-specific, but after injecting a portion of the infectious agent, the portion of that part in our body, specific antibodies will be produced in the body, and those antibodies are going to interact and bind with the specific proteins on the surface of the virus, which are known as the antigen or antigenic determinants. And once the antibody binds with those antigen regions of the virus or bacteria, it's going to kill it ultimately. So that's the job of vaccination. Influence our natural immune system by injecting either a whole virus or a bacteria inside the body or a portion of the virus or bacteria inside the body. Now you may ask a question, like if we insert a whole virus inside a person's body in order to generate antibody, it's similar to that of infection. What's happening in the infection? Persons get affected by a virus, right? So if we inject the virus directly inside a person's body, it's equivalent to just throw the person for the infection. But there is an essential difference between the infections that is done normally and during the production of antibody and the vaccination. Because in vaccination, we not only just throw the complete virus inside the host body, inside a person's body, but in this case we modify the virus in such a way that the virus is no longer effective to cause the disease, but the virus is effective enough to influence the immune system to boost the immunity of our body to produce the antibody, right? So the virus normally has two properties. Remember, let me write it here. It has two properties. One, it can cause disease, manifestation of disease, different symptoms, and second thing, it acts as a immunogen. It has an immunogenic property that helps to boost production of antibodies. These are the two properties that are normally present in this virus. But what we do in the vaccination, if we use the total virus as a vaccine, in that case we should release the disease causing property, we should remove the disease causing property of the virus and only keep the immunogenic property of the virus, so that the virus once inserted, which is in inactive form, cannot cause any disease in the host but can influence its immunity to produce specific antibody that can bind with specific region, in this case let's say the spike protein, and it can destroy the virus, kill the virus at the end. This is the idea behind vaccination for many kind of vaccination that we know. But now, as I told you, there are two ways that we can do that. One way is a subunit vaccine, where we take a portion of this virus particle, any kind of protein we can take, particularly the protein which is very sensitive, which influence the immune system much. So much more immunogenic region, for example the spike protein or the envelope regions or the membrane protein, any of this protein. So if we take any of those portions of the virus and inject it inside to develop the immune system and to produce the antibody, we call it as a subunit vaccine. Other than that, we can produce a live attenuated vaccine. What is a live attenuated vaccine? It's taking the live virus but attenuated form, which we can also call it live killed vaccine. I can say, I mean not live attenuated, or we can also use slash, I can write it killed form of killed vaccine, which is very rare though, where we kill this virus but the proteins that present there still has the property to generate, to influence our immunity to generate antibody. That we can do. Otherwise, we simply inactivate the effect or disease causing property of the virus by treating this virus with some chemical factors or something that will destroy the disease generation property but only keep the immunogenic property. Apart from that, we can also use, so these are the two type, and the third thing is the nucleic acid vaccine that can be done. The nucleic acid vaccine is either we take the nucleic acid of the virus, that is DNA or RNA. In this case, it's positive single-stranded RNA that is present inside the virus. So normally virus, bacteria, eukaryotes like us, everybody has a genetic material. It can be DNA or RNA. In our case, it's DNA. In the virus, coronavirus, SARS-CoV-2, it's RNA. So what we do here is that we can take a portion of this RNA of this virus, and that can be also used to generate immune system, to generate antibody against this virus by taking them. But the use of nucleic acid to produce a vaccine is less effective most of the time. So generally we rely on the subunit vaccine or live attenuated or killed version of the vaccine. Whether we take a complete virus and inject it, or we take a fraction of the virus, a portion of the virus, inject it inside the body to develop the vaccine. So this is the idea of coronavirus vaccine. This is the approach that we are using. Okay, there are many different labs that are doing this work, 40-50 different labs around. I mean, only in the US there are 20-30 different labs are doing. Throughout the globe there are even more labs are involved. I don't know exact figures, so I'm not going to talk about that. In India also the vaccination process has started. There are institutes like National Institute of Immunology as well as National Institute of Virology, NIV. They are also working with this vaccination process. Everybody is given a very steep deadline to produce the vaccine. Normally a vaccine is produced, it normally takes around 1.5 year on average to produce a vaccine, a viable vaccine after clinical trials, and also that we can test it in humans, we can use it in humans, we can market that vaccine as a medicine there. But in this case the deadline is, we cannot afford 1.5 years from now. I mean already 3-4 months are passed, so we cannot afford even 1 year, 2 months, we cannot afford that time. So the deadline was given earlier till November, but all the scientists said it's impossible, literally impossible to produce a vaccine by November. But now we are progressing really fast. We are progressing at an exceptional rate to get a vaccine. But the difficult part is that this SARS-CoV-2, this virus, this particular novel coronavirus strain is mutating very fast. It's very difficult to produce a vaccine for a virus which mutates very fast. What is this mutation? Mutation means any kind of change in this genetic material. So if there is any change in this genetic material, there will be change in the total structure of the virus. There will be change in the proteins that is present on the surface. Now, how our immune system recognizes this virus? Based on the proteins that are present on the surface of the virus, based on these receptor proteins. Now, once we develop a vaccine based on a particular protein that is present in this virus. We take the snapshot, we build a vaccine, and next time if the virus gets a mutation and if the surface proteins are altered, if the surface proteins are changed, then in the next time when the same virus attacks, that earlier vaccine will be worthless. It will not do any work. That is why it's taking so much time to produce a vaccine, because this virus is mutating in a very, very vigorous and fast rate. So keeping that in mind, the CDC is providing the guidelines, and the WHO has a plan to build not only one type of vaccine but building 2-3 different types of vaccines altogether that can all work in combination mode sometimes. So we'll try that in the animals, we try that in humans, but WHO is also been talking about skipping the animal trials and directly giving to the humans, so that we can cut the time to 3 to 4 months shorter, and we can even reach faster to marketize this vaccine, so that everybody can be given with the vaccine. So that is the idea right now. It's going on, but it's mutating very fast. That's a big challenge to produce vaccine against it, but still we're trying it, and you can expect a vaccine by the start of the next year. And meanwhile, there are different drugs, treatments are also being approved. Some of them are not approved, but still they're trying. So everybody is trying their best to control this virus, to contain this virus. But vaccine is the only last way to prevent this virus, because vaccine is something that can protect those who already have the infection, and that can protect those who has not incurred an infection, right? So that's the idea of the vaccine. If you like this video about the coronavirus vaccine, please hit the like button, share this video with your friends and colleagues, and definitely subscribe to this channel to get more and more videos regarding the coronavirus, as well as regarding every single concept of biological subject. I mean literally every single topic, every single subject is discussed in this particular channel. Okay, so that's for it today, and watch in the next video regarding how to protect yourself against coronavirus in this particular time. So that's it for today. Bye.